Hello everyone. Today we're going to discuss the NABL style SOP for serum creatinine measurements. This is a complete functioning and working SOP that is currently being used in a laboratory. For confidentiality purposes, I will not reveal the name of the lab. Any SOP has the same format. Before we get into the details of creatinine, I'm going to explain to you the basic format of any standard operating procedure as required by most NABL guidelines. So first of all, you need a header at the top. As you can see here, you have to have your lab name in a large font size at the very top, and then your address together with the city and pin code. If you go to the bottom of the page, and this is true for any page, you find this footer. The format of the footer is it should first have the lab name and the address, then the name of the document. Every single document should have its unique name. And then we have these 9 details that are required for any given page in any given document, which is basically the date of issue, who has prepared the document, whether the document was amended, the number of the amendment, who has authorized this document, the issue number, the copy number, who has issued it, and then the page number. So you need all these details at the top and bottom of every page. Now this is serum creatinine is basically a biochemistry assay which measures the level of creatinine in the patient's serum sample. So this is usually done either on human serum or plasma, and creatinine usually forms a part of any routine kidney function test. So first of all, we will just go through it line by line, page by page, so that it becomes easy for you to understand. The first thing you must state is the purpose. Here you can see that the purpose is the quantitative in vitro determination of creatinine in human serum or plasma. So what is the purpose of this SOP? It's basically this, which is supposed to be described in 1 or at the most 2 sentences. The next thing is, what is the method? The method is modified Jaffes without deproteinization. So this is the technique that is being used in this lab. You must mention the method or the exact type of chemical reaction that is going to be used for the measurement of the analyte. The next thing that we come to is the principle. That is, it basically details what exactly this method does to measure this particular analyte. So here we have that creatinine reacts with alkaline picrate to produce a reddish color, which is Jaffe reaction. This is a non-specific reaction and is given by many other substances. So specificity of the assay has been improved by the introduction of a kinetic method. However, cephalosporin antibiotics are still major interference. So cephalosporins are basically a type of antibiotics given to patients with bacterial infections, and this may interfere with the reaction. The next thing we mention are the performance characteristics. So this is usually about linearity. Linearity is basically up to what concentration of the analyte can this particular method measure. In this case, measure accurately. So for example, up to a level of 18 milligrams per deciliter of creatinine, this assay is linear. Beyond that level, the assay will no longer be linear, which means that it may either overestimate or underestimate the analyte. Next, we have the limit of detection. That is basically what is the minimum amount or that can be measured by this analyte, and that is 0.08 milligram per dl. Then we have the measuring interval, which is obviously from 0.08 all the way up to 18. Then we have a statement that if the values exceed this range, the sample should be diluted with NaCl or normal saline as required, and the result multiplied by the dilution factor. The next thing we must mention are what are the reagents that are being used, and it's each reagent's concentration. So creatinine by Jaffe's method requires 2 reagents, sodium hydroxide and picric acid. We mentioned the concentration of these two, which is 240 millimole per liter and 26 millimoles per liter. We then must mention the reagents that are used, which is a creatinine reagent, Biorad controls, Herba multical calibrator. Now these are all specific to our method, which we have used in this particular assay. We then mention the equipment used. So what all will be required while processing this sample in the lab. So that is pipettes, sample cups, a barcode printer, a computer. The test in this case in this lab was performed on the EM200, Herba Mannheim 200 biochemistry analyzer. And we must make a statement that the machine will be maintained and calibrated. So that is, we must refer to the SOP for the maintenance of the EM200 and its calibration schedule. Now these are not related to this SOP. Next comes the calibration. So as per the equipment requirement, we do calibration that records are present in respective equipment files. So in this lab, they maintain the records of the calibration of this machine. The Urba XL Multical is used for the calibration. We then mention when is the calibration to be done. So whenever there is a change in the lot of reagents, when the controls are out of range, and when there is any major servicing of the machine, that the machine shows some huge change. The next thing is, what is the calibration frequency? So even without all this, how regularly is the calibration of this reagent done? So the calibration must be performed once per reagent lot using the fresh reagent. So every time there is a new reagent lot, it must be done after 1 month when using the same reagent. So if there is a new reagent but of the same lot, then you recalibrate after a month. If there is a new lot, you recalibrate irrespective of how recent the previous recalibration was. Then if we are using the same kit, that means the same opened bottle on the machine, we recalibrate after 7 days. So we only allow a given reagent to survive on the machine, a given bottle to survive on the machine for 7 days without recalibrating it. And even within those 7 days, if any of the quality control findings are outside the defined limits, the reagent will be recalibrated. Then we must discuss the storage and stability of the reagent. So in this case, it should be stored at 2 to 8 degrees and it should not be frozen. Unopened reagent is stable until the expiry date stated on the bottle and kit label when stored at 2 to 8 degrees. On-board stability is a minimum of 30 days if refrigerated and not contaminated. So that is about the reagent. We then come to the stability of the reagent. So it's just a table talking about this. If the bottle has not been opened and it is kept at 2 to 8 degrees, it is stable up to the date of expiry. If the bottle has been opened and kept at 2 to 8 degrees, it is stable for 30 days. And then some machine specific stabilities are also mentioned. These are probably from the reagent bottle itself. Now the most important thing is, what is the sample type? In this case, we need 2 ml of serum. Next is the type of container that is allowed to be used for collection of the sample. In this case, it is plain vacutainer with or without gel separator or the rapid serum separator. Now, the next thing is the environmental and safety control. So the ambient temperature is maintained in the lab. Temperature should be maintained in collection bag. So when the sample is collected as the test is required, sharp should be discarded. Biohazardous material should be discarded according to the biomedical waste protocol, which will be mentioned in its own separate SOP. It is ensured that all the lab staff are aware about material safety data sheets and are following it. So this MSDS is something which is included in any reagent. Handling of the reagent is done as per the kit insert, and personal hygiene is well maintained and known amongst the staff of the lab. The next thing is precautions. First of all, to avoid contamination, use clean laboratory wares. Close reagent bottles immediately after use. Avoid direct exposure of reagent to light. Do not blow into the reagent. This reagent is only for IVD use, and follow the normal precautions required for handling all laboratory reagents. Follow required safety precautions while handling reagents. So that also includes the safety precautions while handling reagent chemicals SOP, which is a separate SOP. Now, how do you prepare the patient for this test? This is the next thing that must be mentioned. So the first thing is the patient should be conscient. After that, there has to be a TRF or a requisition form or an online requisition before doing this test. The patient should be shown the video about how his blood sample will be collected. This is something specific to this lab. It is not always mentioned. The patient should not be fasting. There is no requirement for fasting. The drug history, if the patient is on any medications, is required only if the patient is taking any supplements. This is especially in context of biotin. The consent, there is no requirement for written consent for this test. Verbal consent is sufficient. Now, what are the limitations or interferences that may be encountered while processing samples using this method? So that is basically hemolysis. Samples showing visible signs of hemolysis may cause interference. Now, is there any checklist before processing the samples? So there are 3 things that this lab follows. Reagents, sufficient reagent for the test is requested, free of contamination, no air bubbles or foam. Control should be in range, and sample there should be a labeled tube received in the required quantity in the right tube and not hemolyzed, because hemolysis can cause interference. Now, how are the results calculated? The machine calculates the results using the following formula. The concentration of creatinine is equal to the absolute of the sample divided by the absolute of the calibrator multiplied by the concentration of the calibrator. So this is not absolute, sorry, this is absorbance of the sample, absorbance of the calibrator, and the concentration of the calibrator. Next is the procedural steps. So the parameter in the EM200 is a machine in which the entire processing, aspiration, dispensing, and measurement is done automatically. So what we have mentioned here is that the parameter is programmed on the EM200 using details given in the kit insert. The samples are processed by placing the sample in the machine, identifying the position and test required through data on the barcode. Again, there is a reference made to another SOP, which is the EM work instructions SOP. Now, the incubation time, the wavelength, the temperature, the absorbance, sample volume, reagent volume are all selected as per the kit insert. The samples are processed by placing the sample in the machine, identifying the position and test required through the data on the barcode. Now, what is the quality control procedure? So there are usually two types of quality controls: internal quality control, external quality control, and the measurement of uncertainty. So first of all, what are the basic quality control procedures? For internal quality control, we run the Biorad level 1 and level 2, 14 days level 1 and another 14 days level 2 control every day. So this is their procedure. For cross-checking of our quality control, we run EQAS, which is the external quality control system. For more details, they have a separate SOP on the EQAS file. After this, we have the internal quality control. So the internal quality control is basically what the lab does internally for its own within the lab. So here they basically have already mentioned that above, but they run 2 levels of the Biorad commercial control are run for 8 hours every day. In the morning, as per the QC schedule, QC is run as per SOP. Again, they have a SOP for running a QC on the EM200. If the QC is not in range, the patient results are withheld, corrective action is taken, and only when the QC is in range are the results of the patients released. We then come to external quality control, which they mentioned as EQAS. So lyophilized samples are received from CMC Vellore. So CMC Vellore is a institute in South India, which is a nodal center for EQOS programs. The sample is reconstituted and run on the machine. So this is the EQAS schedule for biochemistry. There seems to be another SOP for that. We then come to the measurement of uncertainty. Is basically of this parameter is being calculated on a regular basis. The log is being maintained by the laboratory. For LJ charts, we do the entry in Unity. So this Unity is a QC software. Now we come to the biological reference interval for this parameter. So for males it's 0.7 to 1.2 and for females it's 0.5 to 0.9 milligram per DL. We then come to the reportable interval of examination results. So this has been left blank because every lab picks these values either on their own or from a reference. In this case, the reference